So last time we said we've developed a new platform that is a next generation of high-plex proteomics. And instead of being bottom up, it's top down. So we've called this platform the marker platform because really we're using aptamers as surrogates for biomarkers. There are three things that we've done to enable this platform. One is pre-selection. We can identify which aptamers in a library are binding to targets without immobilizing or capturing the target. The second innovation was what we call a closed sequence library. That's getting kind of geometry, but what we really mean by that is that we can use the same sequences in an aptamer library on different samples. We're applying the same samples reiteratively to different things, and we can see then which sequences are working. The third innovation is where we're looking at structure analysis. We'll talk about this in more detail, but really what we've done here is move from sequence analysis to the structure analysis because aptamers work on a structural level. The sequence enables the structure, but the structure is where they work. So this pre-selection is an invention we made and patented called FreeLinks. With pre-selection, the first one, FreeLinks. Pretty simple what we did here, right? But I think kind of cool. We take an antisense for the library on resin. Each sequence in the library will naturally hybridize to this antisense. So we can remove them. The antisense is on resin. We can do a microcentrifugation and remove them from solution. But if a sequence binds to a target molecule in serum or any biological fluid, then it's less likely to hybridize to the antisense. It stays in the supernatant. We just do a microcentrifugation, keep the supernatant, and that's what goes into next generation sequencing or qPCR analysis. That enables us to look at proteins as they are in serum, in complexes with other things, in their native folding. We don't have to do anything to the protein that's changing it. So that's much more complex and deep than what is available now otherwise. The next innovation that was necessary for this platform is what I referred to as the closed sequence space. We also call this a NeoVentures library. What we've done here is we've reduced the number of random nucleotides. We're down now to 12 random nucleotides. The first thing you might say is, that's not possible, you're going to lose too much structural diversity. We've done that by putting these random nucleotides interspersed with fixed sequences. You might say that, okay, that's not going to work, that you're not going to get enough structural diversity. But what we do is we do meta-analysis. We look at thousands of possible templates of fixed sequences in between random sequences. And we find those combinations of fixed sequences, those templates, that give us the highest level of structural diversity. So we're able, on a per position basis, to get as much structural diversity with this as we do in a Celix library, just by screening that. And the only way that that's really enabled is, again, by having closed sequences and being able to reiterate and do the same things over and over. The third one is even that. We're down to 12 random nucleotides. So that means that there are now 16.8 million possible sequences. That's great, because we do each selection now with 100,000 copies of each sequence. And so we can do one round of selection and see how each sequence is being modulated in that round of selection. The problem here is 16.8 million, still a bit of a high number for next generation sequencing. I mean, we want to have multiple copies of each sequence. We just don't have the statistical power for that. So to get around that, what we've done, I think this is the first time, a lot of this is the first time anybody's done this or thought about this, is the third one that is structure. We have predicted the structures of all 16.8 million sequences. We use software for that. We predicted the structures for all. We have multiple sequences that give the same structure. So when we're doing next generation sequencing, let's say we get 10 million reads, we have enough reads per structure that we're getting a reliable statistical analysis on that. So that means that per structure, then we can be looking at averages and standard deviations across samples. And we can do this statistically. And that's what I really meant by this being a top-down approach. Because we apply the same library and we look at everything that's happening in that library. Thanks. I'll talk a bit more about what this means in terms of how this actually helps you. Thanks.